This is the last in our 3 lectures about the microbes of the ruminant hindgut, humans, and other species. And here we'll focus on other species, particularly the horse. By the end of this lecture, you'll be able to contrast the gut physiology and microbiology of ruminants, humans, and especially horses and other species. One review on equine microbiology noted that the microbial community of the equine gastrointestinal tract has received very little attention, despite its importance for the health of the animal and the digestion of feed. And we'll see that on subsequent slides. When I was preparing for these lectures a few years ago, I knew very little about equine microbiology. I thought preparation would be as simple as going to the library and picking up a few books on equine microbiology, but those don't exist. There's surprisingly little known. As you recall, the horse is a cecal-colonic fermenter. It has an enlarged cecum and colon. Fermentation largely takes place in those sites, but as we'll see soon, it takes place in other sites as well. Fermentation begins in the stomach. This is a site that's small compared to the cecum and colon. It's only 20 liters, but it is still a site of fermentation. The upper region of the stomach is non-glandular. It has a pH of nearly 6, and this is the site of fermentation. The lower region is glandular with a pH less than 2, and there's probably little fermentation here. There are 10 to the 8th bacteria cultured per gram of stomach contents. The predominant species that have been identified are lactate producers and lactate utilizers. They're very similar to those in the rumen. For example, Streptococcus bovis or Equinus also occurs in the equine stomach, as well as Megasphaera-like species, which are lactate utilizers. The last slide focused on the lactate producers and lactate utilizers, and it implied that fermentation is mostly to lactate, and we see data here that confirm that point. The figure at the left below shows lactic acid concentrations in gastrointestinal sites of the horse, and the figure at the right shows VFA concentrations. We see that lactic acid concentrations are very high beginning in the non-glandular stomach, indicating active fermentation to lactate. VFA concentrations, on the other hand, are relatively low. They are high only beginning in the cecum and colon. The importance of lactate formation in the non-glandular stomach is that it contributes to acidosis and laminitis. Bacteria are present in the small intestine as well. They're about 10 to the 6th per gram of contents. The groups are probably similar to those in the stomach, though they're poorly studied. Host enzymes in the small intestine have low capacity for hydrolysis of starch. This gives the potential for high input of starch into the cecum and colon, and this is a causative factor in acidosis and laminitis. It's setting up bacteria in the cecum and colon to ferment starch to lactic acid. The cecum and colon are rather large. The cecum has a volume of 35 liters, and the colon has a volume of 80 liters. Combined, their size is about the same as the rumen. In addition to being the place where fiber is fermented, it's a potential site for acidosis, which is causative in laminitis. Despite this, the microbiology is only poorly studied. There are between 10 to the 8th to 10 to the 9th bacteria cultured per gram, which seems to be lower than in the rumen. There are similar lactate producing and utilizing species as those in the stomach, like Streptococcus bovis and the Megasphaera-like species. The cellulitic bacteria are Ruminococcus flavifaciens, Ruminococcus albus, and Fibrobacter succinogenes, the latter two being more minor. And beyond identifying the lactate-producing and utilizing species as well as the cellulitic bacteria, there's been very little work done. Other functional groups have been identified to exist only by selective media, and I'll explain that point on the next slide. This study cultured bacteria on selective media. It examined total cultural bacteria, as well as glucolytic, amylytic, lactate-utilizing, proteolytic, hemicellulitic, and cellulitic bacteria. The fact that the bacteria grew in these selective media implies that there are species that can degrade these substrates, but this study, nor any others that I found, have identified the species themselves. And thus the colon and cecum of the horse is still something of a black box. The horse cecum and colon contains many non-bacterial groups. It contains the methanogens, fungi, and ciliate protozoa. Viruses have not been reported, but most likely they exist. The methanogens are in concentrations only of 10 to the 5th per gram, the fungi at 10 to the 2nd per 10 to the 3rd, and protozoa 10 to the 2nd to 10 to the 3rd. So their concentrations are lower than in the rumen. The methanogen species in the cecum and colon belongs to the same genus as the methanogen in the rumen. The same is for the fungi. The two species belong to the same genera as those in the rumen. The protozoa belong to several families, some of which are shared with ruminants. This slide shows the protozoal families that belong in horses. It's delineated by the green box, and we can see that they have a wide range of morphologies. The blue box shows the protozoal families that are found in the ruminants, and we can see that there's quite a bit of overlap. The families unique to the horses have not been studied beyond identifying them morphologically. It's not clear how similar or different they are from the ruminant-specific families beyond simple morphology. The rest of the lecture will focus on other species, and we'll do it only with a broad view. We'll look at the omnivores, non-ruminant foregut fermenters, cecal fermenters, and colonic cecal-colonic fermenters. Compared to ruminants, there are some similarities. The types of microbes are similar, at least at a rough scale. Microbial counts also seem similar, as do short-chain fatty acid concentrations. There are differences. There are differences in the types of microbes when we look at a finer scale, and the contribution of short-chain fatty acids to energy requirements. This table summarizes the concentration of bacteria, protozoa, fungi, and methanogens across several different animal species, including those that are ruminant foregut fermenters, omnivores, non-ruminant foregut fermenters, cecal hindgut fermenters, and colonic or cecal-colonic fermenters. You can take a few minutes and look at this table, but the next slide will summarize some of the key points. The table on the preceding slide shows that bacteria and methanogens are always present, and they're present in concentrations between 10 to the 8th to 10 to the 10th for bacteria, and 10 to the 5th to 10 to the 10th for methanogens. The fungi are sometimes present. When they're present in concentrations between 10 to the 2nd to 10 to the 5th. As it happens, they're present or probably present in all the species that we saw on the last slide, but it's been documented that some species do not seem to have fungi. The protozoa are sometimes present at concentrations of 10 to the 3rd to 10 to the 6th. Viruses are probably present, but they're usually undocumented, and they weren't in the last slide. Bacteria always seem to be present in fermentative compartments, but there's little similarity in the composition of bacteria across mammalian species, even looking at the level of phylum. The figure at right is from a study that did 16S rDNA sequencing of feces from 59 mammalian species, which shows those wide differences at the level of phylum. In the figure, each column represents a different mammalian species, with the abundance of phyla represented by colored bars. Each color represents a different phylum. You can see that the abundance of the phyla, even the types of phyla that are present, range quite widely across the mammalian species. Culture studies agree with the sequencing studies and suggest little similarity at the species level. Only 14 out of 160 cultured species from pig feces were also found in human feces. For example, Streptococcus intestinalis was the species with highest abundance in the pig. 94% of samples were positive, and it was completely absent in humans. Pigs may be good models for humans in some ways, but they're not good models for bacterial flora. The protozoa are often in different families across mammalian species, but as we discussed before for horses, it's not clear if these differences are morphological or more complex. So we see here again the families that are present in horses and ruminants. They do appear different, but it's not clear if the differences are only skin deep. Fungi are in the same genera as the ruminant, but different species. We can see the genera that are present in the ruminant. We can see the genera for the horse, elephant, and the ass are the same. These fungi have not only been identified, they've been studied, it's been found that there are small but no consistent differences in morphology, life cycle, and physiology. Cultured methanogens belong to the same genus as the ruminant but different species, and we can see that Methanobrevibacter ruminantium is the predominant species in the ruminant. Other Methanobrevibacter species are predominant in the horse, pig, and human. They've been studied, and there are small but no consistent differences in morphology and physiology across these different species. The uncultured methanogens belong to the same groups as the ruminants, though the species level resolution is unclear. And we can see the groups that are present in the ruminant. We saw them earlier. This slide summarizes the concentration of short-chain fatty acids and their contribution to maintenance energy requirements for several species, including ruminant foregut fermenters, omnivores, non-ruminant foregut fermenters, cecal fermenters, and colonic cecal colonic fermenters. Again, you can take a few moments to study the values on this slide, but the next few slides will summarize these values. This slide summarizes visually the concentration of short-chain fatty acids across different animal species and their gut compartments. And we can see that the concentration of VFA are relatively similar across these species, at least in compartments where fermentation occurs. VFA are very low in the stomach or abomasum and the small intestine because little fermentation occurs there. To summarize the values on these slides, the concentration of short-chain fatty acids typically ranges from 50 to 200 millimolar in compartments where fermentation occurs. The contribution of short-chain fatty acids to maintenance requirements is lowest in omnivores, where it's only 5 to 30%. In foregut fermenters, it's higher, between 30 to 80%, and in hindgut fermenters, it's intermediate, between 10 to 40%. Our take-home messages are that the horse cecum and colon are as large as the rumen, but few bacterial species have been cultured. Other animal species have microbiota similar to the ruminant, but only in outline. They have similar microbial groups, concentrations, and functions, but most microbial species differ, as do many higher-level taxa.